So back at it. So hi, everybody. It's really great to kind of sort of see most of you, maybe. Um, I'm really excited to get to talk to you. I was really excited also when, when Chuck pitched the idea of this course. I think it's just really neat to get everybody together to start talking about this topic. I'm sorry that I'm not able to be there this week. It seems like there's lots of great stuff going on, but I will get to see many of you next week. So I'm going to be coming in late Sunday, hopefully by dinner time. So anyway, great to see everybody. Hello, Bruce. Very quickly, I want to just give a sense for who I am and what I do. So my name's Andy Pershing. I was born and raised in Nebraska, which is an interesting place that's in the news for once for something other than football, which I dig. So I grew up in the middle of the country, as far away from the ocean as you can be. My early influences were Jaws, Jacques Cousteau, and my Apple IIe computer. These interests brought me to the East Coast. I went to Brown University in Rhode Island, in part because I wanted to be close to the ocean. And I started doing work in Maine as an undergraduate, doing work on lobster. And then at some point, I decided to move to go on to graduate school, where I worked with Chuck and Bruce at Cornell, studying ecology, oceanography, computers, data, and math. I like to say sometimes that I did data science before data science was cool. And then my research was focused on the Gulf of Maine, specifically the Northwest Atlantic, doing research on copepods like Calanus finmarchicus, doing research on the influence of natural climate variability. So the North Atlantic Oscillation was a big part of our early work. And then translated that into some interesting research on right whale population dynamics that that Aaron has really run with. And I moved to the Gulf of Maine Research Institute in Portland, Maine, and also the University of Maine, where I started doing some work on fish, including cod. And then a really interesting thing happened in 2012, very appropriate for this workshop. We had the first marine heat wave documented for the coast of the United States. So Kathy Mills, who'll be speaking next week, and I wrote the paper documenting the impact of this event, especially the impact that it had on not only on lobster, on the population, but also on the lobster fishery, on kind of the coupled natural and human system at play in Maine. And that got me doing more research on climate change and also got me doing more work communicating climate change. That brought me to where I'm now based at Climate Central. We're in Princeton, New Jersey. Climate Central does climate science and climate communication. I'll tell you a bit more about Climate Central on Friday. And I'll show you these tools that we've been developing, our daily attribution system for air temperature in the atmosphere, and now ocean temperature as well, which is something that I'm hoping to get some folks interested in working with next week. But the reason Chuck asked me here today was to talk about time series. I'm going to talk about time series. I'm calling this time series and me. So it's not so much an overview of how time series analysis works, but it's more just some examples of how I've used time series and some of the toolkit that I think is really can be really powerful. So I have used time series throughout my career, but and I'm going to show you some examples, and I will give you some insights into the choices that that we made as we were pulling these studies together. But I am not a statistician. Right? I'm an oceanographer. I do like math. I like computers, but I'm not by any means a statistician. I do not have a comprehensive survey level understanding of modern statistics. And so that's not what this lecture is. And collectively, I just want to emphasize that you all know way more than I do. Right? Modern statistics and computer science has changed a ton since I was learning how to do this stuff back in the late 90s and early 2000s. My knowledge has evolved some, but one of the things is, I get to hire a lot of scientists these days. And one of the things that I'm really noticing is how great the quantitative skills are among people. So your classmates are going to have a lot of, collectively, a lot of great ideas for how to do the kinds of analyses that you want to do. And I know folks like Aaron have really great background in statistics as well. Okay, but just to give you the idea, so the basics of time series analysis is that we're going to have data. It's going to be based on time. That's the x-axis that I didn't label. So here are two time series A and B. We might have a third time series C. I've labeled these drivers and output. And the idea is that we want to infer a model that in between though, right? And in between them, we have a hypothesis that A and B somehow relate to produce C. And we're going to use our observations of A, B, and C to try to infer how that model works. And I want to point out one other, wrinkle. There's a lot of flexibility in these approaches, right? You don't have to have two on the left, right? You could have just one on the left and one on the right. You can also have A on the left and A on the right. So this could allow you to do things like produce a system that would allow you to maybe make a forecast or prediction about a future state of A. You could also do things like try to just look at the correlations among these, right? Correlation is just a simple model that says that these two things tend to go up and down together. Okay, so I'm going to walk you through an example based on a paper that we published last year with my colleague Adam Kimberlin from the Gulf of Maine Research Institute. This is a paper where we're bringing together a bunch of threads. So some zooplankton data that I'll talk about, some hydrographic data that I'll talk about that does have some bearing on the marine heat wave story. And I'm going to walk you through not only what we found, but kind of a little bit of the logic and the toolkit that we used. And then I'll present a second example at the end that brings us a little more back home to thinking about marine heat waves. So we're going to start with the data. So we have two main sources of data that we used in this study. So the first is a continuous plankton recorder. So that's the little black dots with those colored, the pink and the blue polygons around it. This is an instrument that gets towed behind a ship. They do it roughly every month. It gets towed behind a commercial ship, not a research vessel. So which is why kind of the tracks shift around a little bit. So this is a data set that Chuck and I have been working with for a long time. We were actually some of the first to actually publish on this data. And it's really great. It gives you this really great record of change in the plankton community over time at fairly high frequency, which is, I think, which is really unique in when you're dealing with zooplankton. But then we also have great hydrographic data, and I'm actually going to talk about not the ship-based observations or satellite stuff like Bruce might tell you about. But I'm going to talk about the buoy data that we have in the Gulf of Maine. This is operated by Syracuse, which is part of the Integrated Ocean Observing System. They have a series of 6 buoys in the Gulf of Maine. The buoys have temperature and salinity measurements at different depths. So at 1 meter, 5 meters, 10 meters, all the way down. The one offshore here goes, I believe, 300 meters is the deepest temperature. So we got a lot of data, and that's actually recorded on an hourly basis. But we're going to wrap it up and think of most of our stuff on kind of a monthly or quarterly scale. Okay, so what do we get when we look at this? Well, if we look at the plankton data, we get time series like this. Okay, so when you're faced with a whole bunch of time series, the first thing that happens is your eyes glaze over, right? There's just so much going on in these, in these time series. It's really, really hard to make sense of them. And so the question that we have is, are there relationships among the plankton? So we're not going to think about physics and biology. We're just going to focus on the biology. Are there predictable, consistent, coherent patterns that appear over and over in the variability among these taxa, and are they stable in time? And I want to focus on two, Calanus finmarchicus. That's our big copepod. It's the signature copepod of the North Atlantic. If you put a net in the water and it comes up with Calanus finmarchicus, you are in these, you are in the subpolar ecosystem of the North Atlantic. It is kind of the archetypal species for that ecosystem. So big, abundant, super important species in the North Atlantic. And then Centropodes typicus I'm just going to highlight it. And the reason I'm highlighting it is that it's in the Gulf of Maine. It's probably the most important or abundant species in the summer and fall. So it's really kind of a summer-fall species. And you'll notice that Calanus and Centropodes have sort of different patterns in their time series. So Calanus is high here in the 80s. Centropodes is low. Centropodes is high in the 90s, where Calanus is low. right? It's not perfect, but you can kind of see some patterns. You'll also notice that Entropy's time series looks a lot like some of the other taxa, especially these guys, Parasuto, Withona, Metridia, all have this sort of low 90 or low 80s, high 90s pattern. Okay. So this is the basic data. So the first question we're going to ask is, are these patterns stable? What are the patterns, and are they stable in time? So we're going to use something called principal component analysis. This has been part of my toolkit for a long time. They're great alternatives to principal components, but it's simple, it's easy to use, and I feel like it's easy to interpret. Some of the other things that you might use, like cluster analysis or k-means or things like that, can just be a little bit harder, I find, to interpret sometimes. But the great thing of what principal component analysis is going to do is it's going to look at patterns of covariance and correlation among your data, and then it's going to decompose that variability into characteristic modes. So the way this will work is that we're going to start with our data, and imagine this as like a table that you would have in Excel or R or Python or whatever you're using, where each column is a different variable. So for me, each column is going to be a different species, and each row is going to be an observation of the abundance at different points in time, right? So if I just look down the column, pick a column, I'm getting the time series for one species. I'm then going to try to decompose this. I'm going to pull out one matrix that I'm going to call the modes. This is going to be n by n. So I'm going to have one mode for each species. And for each mode, I'm going to have a number for each species as well. So mode 1 is going to have a number that it assigns to Calanus, a number that it assigns to Entropy's, right, for each of the species that I have in my analysis. And then I'm going to get another matrix that is the same size as my data, that is time by variable, okay? But these variables are not the same. These are no longer species, right? These are sort of smushed together view of the ecosystem. They're the patterns or the principal components. Now, these different matrices that I have a relationship. So just by linear algebra, the modes, if you multiply the mode matrix, or the data matrix by the mode matrix, you get the principal components. But there are lots of matrices that you could do this, right? You could have the identity matrix here, just like zeros with ones on the diagonal, and then you would just get your data back out, right? There are lots of matrices that would do this. Our mode matrix, right? Our principal components are special in that what we want is, when we do this, that mode 1, in this case, the column on the left in my principal component matrix, has the maximum amount of variability that we can. We're going to take all of as much of the variability from our data set, and we're going to try to stuff it into that first mode. And then we're going to see what's left and try to stuff that into the second mode, see what's left, stuff that into the right mode, right? So we're going to have decreasing amounts of variability or variance explained as you move from left to right in the principal component matrix. And the way this relates back to the data is so if we have a column here, right, this is one principal component set of weights, right? It's a weight. This is a vector, an n by 1 vector, right? We have a value for each of the variables in our data set. This vector is going to give us this principal component. And we're going to have a mode here. It's going to give us to this one. And so the idea is that each mode, right, my orange mode, right, we're going to multiply essentially what happens is that principal component 1 over here in our matrix is this vector. It's the data weighted by this vector. It's a weighted sum of the data, okay, weighted by the values in the modes, okay? Lots of, every stats package that you can have in R, Python, MATLAB, whatever you're using, will have a way of getting these values. There's some different tips and tricks that you can use to try to get them out, especially when you're dealing with gaps in your time series or things like that. But all of them, the basic idea is that you're going to take your original data and you're going to decompose it into these modes. And both the modes and the principal components are now a resource that you can use to try to understand the system that you're studying. So for me, when I do this with the zooplankton data, I get these two modes. So I actually get more modes, right? I have 7 species, so I'm gonna have 7 modes. But I'm only gonna talk about the first 2 in this case. Together, these first 2 explain about 81%, I think 81% of the variance, something like that. And the first mode explains more than half the variability. The second mode explains about a little more than a quarter of the variability, 28%. And what I'm showing over here is if you take that vector and you just put a bar graph on top of it. So I'm graphing the first column in my mode matrix. Those are the orange bars. So you can see that Calanus here has a negative value. Centropodes has a big positive value. Oithona has a big positive value, right? We have these species here in the middle. We're giving them a lot of weight, and we're not giving a lot of weight to the others. So that means that most of the variability in my data set is captured by essentially the mean of these 4 species. If you just take the mean of those 4 species, you're getting, or those 4 taxa, you're getting about half of the variability in that time series. Then mode 2, we essentially, we ignore these, right? These guys are getting basically close to 0. And the big thing is that we're putting a lot of weight on Calanus, and we're putting a lot of weight on some of the other larger species as well. So you can think of it as, in some ways, sort of a large versus small relationship. Okay, so then you'll notice up here that I labeled this 1961 through 2003. That's because Chuck and I published a paper way back in 2005 that used this same data and came up with this mode. And we're just kind of highlighting that modal pattern. So the question is, if we add now another 8-9 years of data, how does that change our view of the system? Because over this time, 1960 through 2003, the Gulf of Maine actually hadn't warmed very much. It was a fairly, temperature was not the big story. But after 2010, temperature really became the story. And so that's one of the questions that we're asking. So here I just add, here we're looking at 1961 through 2017, so the whole data set. And here we're looking at just the last 17 years, 2001 through 2017. So you notice that the pattern stays the same. So we're seeing that there's this fundamental relationship that doesn't really matter which period of time we pick. We see this common set of relationships that when Calanus is down, Centropodes is up. When Centropodes is up, the other small species are up as well. Okay, and lots of ways you could think about interpreting it, but I really feel like this is a fundamental property of the North Atlantic plankton community. We do see similar patterns if we do this analysis in other parts of the ocean. And one thing I want to highlight while I'm using annual data here. This looks a lot like the seasonal cycle, the seasonal shift in the community, where we go from a spring plankton community that's dominated by Calanus to a summer community where it's much more diverse and where these other species play a bigger role.
Andy?
So there's a chat, yeah.
So one of the students asked, what does it mean that the PC weights are positive or negative?
Signs in a principal component are arbitrary. So they're important relative within a mode, but they're arbitrary in terms of which across the modes, right? Because we're looking at variance, which is scaled. So we just wanted to do this to highlight. We took the first mode and we made sure that the sign of the first mode in all of these had Centropodes being positive. And then we made sure that Calanus had the same sign when we were graphing mode 2 as what it did in the first mode. But you could flip it around if it made more sense. But the sign is arbitrary.
Okay, thank you.
Yep. All right, great question. Okay, so what do we find? So now we have these vectors, right? We have our 2 modes, and now what do our principal components look like? Well, they look like time series, right? Because we're going to take a vector, we're going to multiply it by our data set, we're going to get a weighted sum of our data set, which is going to be a single time series for each of our modes. So mode 1, I'm plotting here in orange. You see it has the low in the 80s, high in the 90s, low in the 2000s, looks a heck of a lot like the centropies graph, which I'm sure you all remember clearly. And if you look at mode 2, it looks a lot like Calanus. And here I've made sure that I've adjusted the sign so that it looks like the Calanus, so that it now the sign of the Calanus one looks like the Calanus data set, right? So you see that they're offset, right? Low in mode 1, high in mode 2, high in mode 1, low in mode 2, right? That we're getting a little bit of a flip-flop going on between these. And then we also have these very identifiable regime shifts. So we ran a regime shift detector. It identified, I forget the years, 82, 89, 2001, 2000, I think 12 here, 11 or 12. And anyway, it sort of, the Gulf of Maine seems to go into these like very stable states where the system, especially that mode 1, it gets sort of locked in on that pattern. And so the big question is, well, what's driving those shifts in the system? Okay, so we're going to bring in the hydrography. So we only have the hydrographic data starting in 2002, but we have a lot of it. And so what do you do when you have a lot of time series? Throw principal components at it. At least that's what I do. And so the question that we're asking is, do the physical changes explain the plankton shifts? And so we're going to run a principal component analysis on the buoy data. And so each panel here is the buoy B, E, F, I, M, and N is the offshore buoy. Top row is salinity. Bottom row is temperature. And then within each panel, we go 1 meter, 20 meter, 50 meter, 100 meter, 150 down to 180 meters, which the coastal buoys don't go down to 180 meters. Okay, so we've got a lot of data, but the principal components help us make sense of that data. So mode 1, which are the orange bars, you'll notice the orange bars are positive up here and are positive down here at all depths. And what that means is that basically, to a first approximation, this region responds as a unit. So if it's warm, it's also salty. If it's cool, it's also fresh. And if it's warm and salty in one place, it's going to be warm and salty everywhere at all depths. Okay, so everything's just sort of going up and down together. Mode 2 gets a little more interesting. Mode 2, you start to get some contrast. So you see that it's fresher in the it shows, sort of fresher in inshore, saltier offshore. But the big contrast is that it's warm everywhere and also salty offshore. Okay. And so we get now these two time series. So now that we have our time series, now what we want to do is try to make sense of how the time series relate to one another. And for here what we're going to do is just a lot of just correlation analysis, but shifting around in time, right? Because of cause and effect, if something precedes something else, you have an idea that that's potentially a causal relationship. Oh, just want to highlight the 3 heat waves, 2012, 2016, and I think this one's picking up 2018. Okay. So how do these hydrographic modes relate? So I'm sorry I didn't have time to animate this one, but what I'm showing here is the driver buoy principal component 1 correlated with buoy principal component 2. So I'm looking at buoy principal component in quarter 1 correlated with buoy principal component in quarter 1. What's the correlation? 1, right? Anything correlated with itself, it's going to be 1. So you're going to get a diagonal. But you also see that buoy quarter 1 is also correlated with quarter 2, quarter 3, quarter 4, and it sort of declines down as you move in time. So that's autocorrelation. This shows persistence in the system. In buoy 1 seems to last more or less a year, and you see that going through. And so you see then also correlations between quarter 2 correlating with quarter 3 correlating with quarter 4 correlating with quarter 1 the next year. Okay, a little bit of a reset around the end of the year. Similarly, if we look at this panel down here, the lower right panel, this is buoy PC2 correlated with buoy PC2, same sort of persistence and same autocorrelation structure. What gets, I think, really interesting though is that buoy PC2 precedes buoy PC1. You see this sort of big red blob right over here. What this is saying is that offshore happens before inshore, right? If we get a change in that offshore buoy, which is really what PC2 is, then we see that propagating onshore the next year. So there's a little bit of predictability in the system. And kind of what we're seeing is that offshore pattern can come inshore. Okay, and so this is very consistent with some of the, some papers that, that Chuck and others have been publishing about how kind of the offshore, the water that's coming into the Gulf of Maine sets up the dynamics, especially it's sort of a preconditioner for marine heat waves. So the basic idea down here on the bottom is that if it's warm and salty offshore, then the next year it's going to be warm and salty inshore, right? That signal is going to move inshore. And that's going to be a preconditioner for these marine heat waves. Okay, we can also do sort of similar sorts of correlations now with our zooplankton. So here's buoy PC1 as a forcer on Centropages and as a forcer on Calanus. So buoy PC1, when it goes up, Centropages goes up. So warm and salty leads to more Centropages. Warm and salty leads to lower Calanus, especially in this in Q3. Okay, so if we move down a row, Centropages highly correlated with itself, as you would expect, right? They live for for a month or so that you're going to get some persistence. Centropages does not predict anything about Calanus. There's nothing significant going on here, really. Calanus, on the other hand, does predict Centropages. So if Calanus is low if Calanus, excuse me, is high in Q3, then Centropages is going to be low in Q4 and basically for the next year. Okay, so you get this switch in the system where Calanus sort of seems to be a little bit of the trigger in Q3 that then can set up this Centropages pattern that persists. And you notice this is where we see the correlation between the buoy and Calanus. So the idea, kind of the model that I'm sort of proposing here, is that you get a shift in the buoy, it leads to a shift in the hydrography, leads to a shift in Calanus in the summer. Let's say Calanus goes down in the summer. That then allows Centropages to go up in the fall, and then that persists. So why might that happen? What might be going on here? How can you get this persistence? Well, in the paper, we hypothesized that it's predators, and I didn't emphasize that time series, but we see that invertebrate predators like Keating gnats are also very abundant during these small copepod or Centropages regime shift modes. So the idea is that if you have cool condition or mixed condition that occurs in either the spring, characteristically the spring period, or the early 2000s, you get a lot of Calanus. That leads to this negative correlation with Centropages. So Calanus sort of keeps Centropages down. Because you have less Centropages, that means you're going to have probably fewer predators. And so that allows Calanus to stay big. So a little bit of a feedback loop that we're hypothesizing. And then over here, if you shift into a warm period like we have in the 2010s, or a stratified period like we had in the 1990s, the stratification there was not temperature, it was salinity driven. That the stratified period leads to lower Calanus, so more stratified leads to more of these small guys, leads to more predators, which leads to more Calanus, which leads to fewer Calanus. So potentially a feedback loop. So that's sort of our synthesis of that data based on the principal components analysis and then based on the lagged correlation analysis. All right, so that's a lot. Hopefully the biology makes sense. I'd love it if folks thought the biology was cool, but for the purpose of this class, it's really like the logic of the time series of taking a whole bunch of time series, using principal components to like compress it down into a manageable number, in this case, these two modes, and then starting to work through the logic of what happens first, what happens next, using the lagged correlations. Okay, I want to show one more example, and this is from a paper that I led in 2018, looking at the 2016 heat wave in the North Atlantic. And I want to just highlight that the title of the paper was Evidence for Adaptation. So keep this in mind as you're thinking about definitions of heat waves and what it means, especially as you're thinking about the impact of heat waves on biology or the impacts of heat waves on a human system. And so this is really building on or following up work on from Kathy Mills' paper on the 2012 heat wave. So why did we do this study? Well, we did this study because in 2016, we had a second extreme heat wave in the Gulf of Maine, and the temperatures looked a lot like the temperatures that we had in 2012. So 2012 is red, 2016 is black. So there's some interesting differences. In 2012, it was a little higher in the spring. 2016 was a little higher in the late summer, but overall, pretty similar years between both of them. What we saw in 2012 that Kathy wrote up in her paper was that the marine heat wave led to earlier lobster landings. So lobster in the Gulf of Maine gets active as the waters warm up. As the lobster get active, they get easier to catch. And so you end up with lobster landings basically following the activity level among the animals. We also saw in 2012 a decline in lobster price. And that decline in lobster price led to really bad vibes in Maine. It made people super, super grumpy. It almost caused a border skirmish between Maine and Canada. Because at the time, a lot of the lobster that was landed in Maine had to be shipped to Canada to process it. And the Canadians were like, don't send us any more of your nasty American lobsters. We're done. Like, we have all we can hold. And so it was really kind of an interesting event. 2016, we have similar temperatures. So what happened to the lobster fishery? Well, first thing we're going to do is we're going to look at lobster landings. So lobster landings, you can see, vary from year to year. Each curve is a different year. I've highlighted 2012 in red and 2016 in black. Similar temperatures, similar patterns in landings. In both 2016 and 2012, it was early. So especially you notice what happens in June, this uptick in June, where June landings are comparable to what people would normally expect to get in July. And then overall, just really, really high. And the absolute peak here reflects both the activity, but also just more lobsters in later years than there were in some of these earlier years. Okay, but what about prices? So prices are really interesting. So 2012, here's that collapse in price that made people super grumpy. June and July, too many lobsters are coming in, not enough places to put them, so the price went down. In 2016, though, the prices are much more stable, right? They were high in 2016. So question that we asked was like, how much higher? Like how different were they in 2016 through 2012? And that requires us to figure out, like, what would we expect lobster prices to be? So the way we approached this was to model the price of lobsters as a function of, first and foremost, just the previous price. You assume that if the price was high in the previous month, it's going to be high in the next month. So that's autocorrelation. But we also are considering whether landings have an effect, right? Supply and demand. If you have a lot of supply, you would expect that the price would go down. And so we tried a few things, but what we came up with was this kind of relationship, so that the price in May is a combination of some sort of weighting of the price in April and a weighting of the landings in May. And B is usually going to be negative. So if price is high, if landings are high, price goes down. Okay. But we then did this for each month. So we did it for June. We got a June relationship. Oh, I didn't change my subscripts there. Sorry. We did it in July. And then we also did it in August. August, we did not find a significant relationship with August landings or with landings for any month. It just seems to follow what happened in July, at least according to the statistics. And here we're just this is just linear regression, but it's linear regression sort of playing around with the lags in the variables. And then we built a model for September, and I think we continued it out maybe until November. Okay. One of the things I want you to notice is that how the output from the one row can become part of the input for the next one. So for example, if you give me April prices and May landings, I can estimate a May price. I can drop that in here. If I have June landings, I can get a June price, right? And so I can sort of follow that through. So what we did was we simulated 2012 prices using the April price in 2012 and then the observed 2012 landings, and just sort of walked through this process. We had some Monte Carlo action going on there, drawing kind of different models and fitting things there so we get some variation. But the idea is that we're going to simulate 2012 using only the landings, right? And what would the price be? And we're going to repeat this for 2016 using 2016 April price and April landings, right? And so we're going to get now a simulation of what 2016 would have been using kind of historical relationships between landings and prices. Okay, so this is our time series. And this is what we suggest should have happened in both of those years. So it should have been a little higher in 2012. I'm sorry, in 2016, the black line here, than in 2012. But you'll notice then that the anomalies are really different. That actually the 2012 just, tends to follow the historical patterns pretty well. So, the decline in lobsters in some way, the decline in lobster prices in some ways just reflects a lot of landings, right? A lot of lobster coming on shore, price goes down, but there's a little bit more of an anomaly. Overall, if you integrate that red curve, you get $38 million total of landed value for the fishery. On the other hand, 2016 is really, really weird. 2016, you get this big boost in price, much, much different than what you'd expect based on the historical relationships between price and landings. And so we interpreted that as a sign that the lobster fishery had changed its behavior. And in particular, we think it's related to the marketing and processing of lobster. So following 2012, everybody remember bad vibes in Maine, everybody was freaked out. They got money from the state, they got money from the federal government, they got money from the industry, started a big marketing campaign, tried to bring some of the processing back to the United States from Canada, which is where most of it was taking place. So now you have just this greater ability to deal with the higher volumes of lobster that they saw in 2012. And when it happened again in 2016, they were able to capture a lot more of the value of the fishery. They sort of broke that relationship. Okay, so quick conclusions. Time series analysis is fun. You can build a whole career on it. There's lots of stuff that you can do with a simple toolkit. 98% of my papers are principal components analysis and regressions of one kind or another. Occasionally I do some differential equations, and then that makes me really happy. But a lot of the work I do is just sort of these two kind of simple, fairly simple toolkit. And then just my last bit of statistical wisdom here is don't rely on statistics. I think we always kind of want to be able to find like the new tool, the greatest thing, like machine learning this and AI that, but don't rely on it. These are not going to be able to, well, forget AI, maybe it can do it, I don't know. But at least, a lot of these complicated tools, most of the time, they're just adding a little bit of value on top of what you could get with some of the simpler analysis. It's really more about the processes that you're trying to study and the hypotheses that you want to test. And really, like, think about the data, think about what the data represents, and then go to the statistics. Rather than, I would advise you, rather than like trolling the R libraries to find whatever the cool package is of the moment. Okay, and that's all I got.
That was great, Andy. So why don't we go around and people have
Yeah. Yeah. No, I said another one. I also have another one in what are you talking about? Andy, can you see my question in the chat?
Sure, chat. Let's see. Okay, can you please explain again the steps of how you get from the time series of the two modes, the blue and the orange line, to the correlation heat map? So now I get the math behind the step. Is it just for each quarter of the year, the modes correlated with the other quarters? Did you also try other time frames? Yeah. So thank you. What I'm doing in those plots here, I can zip back so we're all talking about the same thing. Is what I'm doing here is we have our quarterly time series. So for the hydrographic data, we broke it down into quarterly resolution, and then what we're looking at here is how the quarter one relates to quarter. For example, in this cell here, how quarter one relates to quarter one for principal component one. So it really is just like I take those two time series, PC1 quarter one, PC1 quarter two, and I'm just correlating those together and showing that it's roughly, I don't know, 0.75, something like that. And then down here, what I'm doing is taking, for example, principal component in quarter three and correlating it with PC2 quarter three with PC1, for example, in quarter two. So from the next year. So I'm just kind of shifting these time series around in time in order to kind of highlight like where do we get relationships. And visualizing these sorts of correlations, I find to be really really fun, and I have not found a way that works for everybody. So this was like the way that I came up with that like made sense to me. It didn't manage to make it through review, so I guess it made sense to at least one other person, two other people. But these sorts of relationships, I find especially with lags, it's really hard to kind of keep them straight, and it can be really tough to visualize them in a way that's kind of obvious. You do have to kind of sit with this sort of data a little bit. Okay. Thank you. Somebody agrees with me that AI is just fancy regression.
And did you also try other time windows or like time frames, not like quarters, but like other?
No, we didn't. I mean, we kind of felt like quarterly was about we wanted to be looking at change within the year. And we didn't feel like we could, especially once we wanted to bring in the plankton data, that we could go much finer than quarters with the temporal resolution of the plankton anomalies. So quarterly was sort of right for us. I've done quite a bit with the annual scale data. That's a lot of the earlier stuff that Chuck and I were doing. We were making that with kind of like an annual roll-up of data. I've moved a little bit away from that just because, I think there's so much interesting stuff that's going on within the year, especially in the plankton community, that I've kind of been interested in trying to understand that.
Okay, thank you.
Yeah.
Hi, Andy, I have a question. I think it was within the last couple of slides just before this one. It was the time series that you had for the principal components. I'm wondering, you mentioned that it was indicative of some stability over time with respect to those species that you picked. I'm wondering if you can explain, like, if you didn't see any stability over time, would you kind of go about the same methods that you did and still doing that principal correlation or principal component analysis correlation, or would you do something else, or what might you infer from that?
Yeah, I mean, so I think, are you talking about this one where like, okay, if we did the PCA back in the dark ages, we got one relationship and then we sort of show that relationship is still around. Is that kind of what you're? Yeah. It's a good question. I think that if we had, I'm not quite sure what I would have expected. And I guess we talk a little bit about this in the paper, that in some ways you expect like the plankton community to have these sorts of logical, essentially like a logical seasonal, seasonal cycle, and that if you perturb it in one way one year, that you would get like the same response in the next year. What I'm kind of interested in is at some point this will break down, right? And at some point we will do this relationship and we will see something different happening. And that's going to be a signal that the Gulf of Maine plankton community has fundamentally shifted, that basically we've warmed it so much that we're dealing with maybe more of a temperate community, or there's some other shift in the system that causes that change in behavior.
Thank you.
Hi, thanks for this talk. I had a question about your model looking at the relationship, or I guess the predator control of chaetonaths on Calanus and Central Pages.
Yeah. So
Okay, so the question is this. Sorry, there's a lot of feedback here. Okay.
Yeah.
So the general question is, I don't quite understand if the chaetonath predators are only predating on Calanus, or if they're also predating on the other copepods. And if they're also predating on the other copepods, why would there be a higher abundance of predators in warm stratified conditions versus cold mixed conditions?
Yeah, great question. So there's a lot going on here, and this is where I'm bringing in a synthesis. So not all of this is in the data. Some of this is in other literature. But the basic idea is that within the seasonal cycle in the Gulf of Maine, in the spring, when Calanus is the dominant species, the water column is very mixed, much more mixed, and starting to stratify. You get the spring bloom. Calanus is the spring bloom specialist. It has evolved to be able to take advantage of that pulse of productivity. And then Calanus goes into diapods. Not every individual, but many of them will go into a state of reduced activity at depth, a couple hundred meters below the surface. They're just hanging out there, living off of a lipid reserve. One of the questions people have always asked is like, why does Calanus do that? Like, why does it go through that diapause period? The main hypothesis, or the hypothesis that I tend to favor and that I think has the strongest evidence, is actually a predation avoidance hypothesis. Calanus is much larger than the other species that I was talking about, except maybe for Matridia. But Calanus is a much larger species. It takes, in order to be big, it has to have a long development period. And so that means that over the course, that means that it's more sensitive to predation than the smaller, shorter-lived species. And so what I'm showing here is almost more like the sensitivity to the predators. So the idea is that if Calanus, in the conditions when Calanus is really abundant, in the spring or in the early 2000s, the hypothesis would be that the predators are lower, have less abundance, lower abundance, probably, averaged over the year, but and they're doing that by having a restricted period of time where they're abundant. And that means that they have less control on Calanus, and then Calanus can kind of muscle out some of the other species. Whereas in the stratified period, Calanus does not like stratification. Part of the reason why it doesn't like stratification, we think, is because of the predators. And so we think what's happening is that Calanus, in the stratified conditions, you build up this, you shift into the summer mode, you build up this population of other species. That allows the predator community to get going earlier. It allows the predator community to get more, to become more abundant. And then Calanus is just more sensitive to that than the other species are. But this is hypothesis land here. We really are kind of putting this out there as one explanation that we think can explain the stability of the patterns between the decades, even though the forcing is really different. and some of the features, the persistence that we see, the reason why we get locked into these kind of regime shift-like patterns in the Gulf of Maine. Some of it's probably the kind of persistence in the physics. We think some of it is also the kind of the persistence and the feedbacks in the biology. And so the hypothesis, you're all in Friday Harbor, so hopefully you get the analogy, but we're really thinking that, Calanus is in some ways, Calanus is the dominant competitor, and that Keytig gnats and these other invertebrate predators are the keystone predators. They're like the sea stars of this system. That's kind of the hypothesis that we're putting out there.
That's cool. Thank you. Thank you.
So Andy, let's see if can you hear me, Andy?
I can, yeah.
Okay. So one of the things that I think is important when looking at this figure, if you remember, if you look at the time series of Calanus for the late stages, it's very different than the Calanus, the early copepodite stages, which kind of line up more with the smaller copepods.
Yeah, they tend to fall with them. They are Calanus 1 through 4 does have some action with that first mode. Yep.
So go back to the other slide that you were looking at. So what I'm thinking is, especially during the summertime, Calanus the population is really dominated by the younger stages, right? So they are going to be more vulnerable to the predators. Whereas if you look at at least in the early spring and throughout much of the spring before the population really starts, going strongly, those are the C5s, the C6s. They're not going to be very vulnerable, at least to the invertebrate predators. So that would be part of the explanation, is where you are in the life cycle of Calanus, and how prone you are, at least to those invertebrate predators. I always have some concerns about interpreting what's going on with the predators, because certainly the fish are responding different. The Calanus in the different stages are responding to fish differently than to invertebrate predators. The other concern I have is that the invertebrate predators that we get in plankton nets often don't include things like the like the Nenomia, the siphonophores.
Yeah.
And those are often really important predators, and we just don't sample them well with nets. So I think predators play a big role, but we're missing a lot of the important invertebrate predators in the samples that we have.
Yeah, no question. And I think, sorry, just a little feedback. We looked at, I won't flip back because I think people are probably getting dizzy, but the Centropodes time series, you see what Chuck was talking about, where it's basically like zeros and ones. It's like either the CPR catches one or catches nothing, or it catches a bunch. And we do see kind of more centropody, or more of, sorry, not centropodes, more of the Keytig gnats. We do see more Keytig gnats in the CPR data during these low Calanus, high centropodes periods, like the 90s and the late 2000s. But it is by no means as nice. I'm less confident in those time series, which is why I didn't actually include them in the principal components. They were sort of brought in as an explainer after the fact.
Got a question from Bruce.
Oh no.
You can hear me okay, right?
I can.
Okay, I'm thinking about just a technical question, I guess. When I do satellite stuff and use principal components, and if I'm doing it with two different parameters like SST and chlorophyll. You can divide by standard deviation. I wondered if some of these populations of different species are going to be going through wildly different numerical, abundances. And in the case of satellite data, you'd like to divide the temporal standard deviation by each respective population. I wondered if you did that, and or if you use the biomass instead of the numerical abundance to compare, species 1 to species 2, et cetera.
Yeah, a great question. So Bruce, the way we've done the CPR analysis is we're expressing it as essentially standardized anomalies off of the annual cycle using like a log, kind of a log off of a log transformed annual cycle. And so we're looking at essentially log anomalies and then averaging those up, and that's what our index is. And so that, because we're kind of pulling out the annual cycle, you are removing the fact that like Calanus could be, sometimes an order of magnitude more abundant than the others. It's certainly, actually, no, I'm going to confuse myself here because we're talking abundance versus biomass. Calanus might have a couple orders of magnitude more biomass, but it can actually have lower abundance than some of those other species. And we have to deal with the CPR that the CPR is sampling the world through like a 1 inch square aperture. So it's not the best way of sampling plankton. And so we really are trying to look at each species or each taxa relative to itself. And so we are doing that standardization that you're talking about. So it's really, really important when especially if you're bringing two different data sets together. If one data set just has more variability than the other data set, or one variable within it has more variability than the other, the PCA is going to really like lock in on that one. And your first mode is going to be basically like plucking that one out, or plucking that, it's sort of separating your time series, where if you're doing like a joint SST chlorophyll analysis, you want to understand kind of the connection between the two. So you would really want to make sure you're looking at like standardized anomalies, dividing by the standard deviation, making sure that everything is, normally distributed more or less, so that everything is going to kind of play nicely with itself. So you're all muted.
Okay.
I dropped, there's some chats questions, and I put one in there, as did Zena, I think in attempt to reduce the feedback. Thank you, Andy.
You're welcome. Let's see. Oh, good, a lobster question. Let's see, regarding the plot with lobster increase in 2012, there's another gray line which is unusually high and peaks early. So also due to high water temperature. I don't remember. In general, would you say lobsters, and I guess lobster industries, would benefit from marine heat waves? So I'll answer the last question. So the lobster industry in Maine, I would say, did not benefit from the first marine heat wave, right? They lost $38 million. The losses are probably higher than that, especially when you factor in, fuel prices and other things that year. I would say they then were able to benefit the next time the heat wave came around, because they basically created a system that could take advantage of kind of the heat wave state, or the warmer conditions. But that's, I think what's really interesting is humans are really adaptable. Like one bad event scares people, they tend to like try to avoid it the next time, you hope. And it looks like the lobster industry did that. But think about, the impact of the heat wave on the lobsters themselves. Think about like how the lobster population, responds to a heat wave versus, a plankton population that's, turning over at a much faster rate. Compare that to something like, Arctic Icelandica, like a 200-year-old clam living at the bottom of the ocean, right? Those are all different timescales. And I think this is where like, there's a really interesting intersection with the debate that I'm sure you all will get into about kind of the definitions of marine heat waves and should you standardize or detrend or keep your baseline stable. I've tended to think that, the really interesting thing is that, the heat wave is not a property, right? The temperature is a property, and that's going to interact with the with the biology or the process that you're trying to understand. Those processes are going to have a particular time scale with which they can adapt or adjust to the environment. And so you need to think about how your definition of heat wave is going to align with that scale. All right,
Great,
so
thanks,
folks,
Andy. I just had a quick question. Earlier you'd mentioned that these analyses are appropriate for large time series. Could you specify a little bit more what that would be? Is that decadal level time series? Thank you.
I was thinking large more in terms of the width of the matrix, in terms of like the principal components and like how many different time series that you have. So principal components is really a way of like trying to, compress your data down into a manageable level that you can then start to analyze in a way that, that the human brain can handle. So depends on how big your brain is. Mine can kind of deal with 2 or 3. Some of you might be more multidimensional than I am.
One quick question.
Okay.
Sorry, Andy, me again, Bruce. Were there other fisheries besides lobster fisheries in Maine that did better or worse during these heat waves?
So the interesting thing about the lobster fishery is that it's the regulations are basically like, do you have a pulse in a boat, right? I mean, there isn't like a quota or anything that constrains it. It's really just like who has a license. And once you have a license, you can go catch as much as you want. You're restricted to the number of traps. So it's an effort controlled fishery. And because it's a fishery that essentially operates year round, it reflects the biology, we think, more than some of the other fisheries where when you fish and how much you catch can depend on a lot of factors like quota and fuel prices and prices on shore. So there's less of that going on in the lobster fishery. In 2012, within the year, the lobster was the really big story. In subsequent years, we realized how, that was a really bad recruitment year for cod that then led to some interesting management challenges that we wrote about in some other papers around cod. It was also a really interesting year, I believe in bluefin tuna. I think that was one of the years where they found like bluefin tuna off of Greenland because the water was so warm that the fish could, just really track that. And we saw some of that tracking. I think squid is the other species where we saw a really strong response. And I think that's kind of an interesting global story that like squid are just so able to really, really react quickly to changes in temperature. So, Humboldt squid moving north during an El Nino year off of California, or I believe there are some squid dynamics off of Tasmania and some of their heat waves. So I think squid are kind of a really neat species.
Okay. Thank you very much, Andy. I think what we should probably do is like take a 10-minute break, try to resolve a few more audio visual issues. And so why don't we come back at 10:15?
Okay, and I will see you all, I think, on Friday. And if you have additional questions, please reach out by email or Slack. I'm on the Slack system. So
Great.
all right.
Thank you very much.
See you later.